This is the same picture, but only leaves were removed before picture was made. So we control flower bud blasting observed. The flower is dried. Gibberellin A3 grows of the stems, but here no growth. When we determine we measure the length, so GA in February 8, stimulatory effect, and February 24 growth is much higher on the treatment. We measure the leaves, so generally speaking, as of the leaves, we have first leaves, second leaves, and third leaves. Measure February 8 and February 24. Generally speaking, the growth of the first, all the leaves is stimulated by gibberellin. This number, the second one. Similar experiments we made with cool tulip bulbs. After cooling, again was removed the root primordia, and we kept in the water GA3 ABA and GA plus ABA, GA at concentration 200 milligram per liter, ABA and 20 milligram per liter. You can see that in this case also that growth is stimulated by GA. So next step, later stages GA3, and here we have ABA also have some inhibitory effect, inhibit GA action. The next stage, control over floweret. This is mid time of flowering. Again, the growth of different after this treatment, GA highly stimulated the stem growth. So the growth, after cooling, the growth is not stimulated after cooling in different leaves, you can see, but ABA inhibited. We know also that we have these kinds of artificial substances like paclobutrazole, which inhibit the biosynthesis of gibberellin. So if we applied as a solid range paclobutrazole, or we applied paclobutrazole and treated with bulbs were treated with paclobutrazole, we have inhibitory or stimulation. If gibberellin is production is inhibited, we have smaller growths of the stems. So this kind of paclobutrazole is very often used in physiological studies. So we can see that all time we have, from one hand, we applied on the top of auxin, we have observed stimulatory growth of the stems. On the other hand, if we apply it to the bulbs gibberellin, we have stimulatory effect observed after treatment with gibberellins. So problem is how to combine the action of two different hormones. I will do in the later stage of my talk. In next our study, our question was, what about the effect of auxin on tulip stem segment excised after planting tulip bulbs? So in this case, after planting of fully cool tulip bulbs, when the stem of length was about 6 centimeters, we isolated these stems. We cut near the basal plate, and all leaves was removed, and the flower buds was removed. It is control plants, and this explant were kept in the water only, and on the top was only lanolin paste. But here, also was the base of the explants were kept in the water, but on the top was applied IAA. So you can see, if we use only explant without tulip bulbs, we can induce stem growth. During 7 days, we have growth, but here no growth. So some endogenous substances present inside of the segments it is enough amount is present for enderogation to this degree. Here it is similar. We use here the longer explants when the length was about 12 centimeters. If I applied auxin on the top, we have also elongation of internodes. So we can see that that auxin is also responsible for the elongation in the explant, isolated from cool tulip bulbs. Here is the dynamics. Previous experiments in the after 8 days, the control no growth, but the growth start after 2 days. You can see observed similar effect when the length of initial was 6 centimeters. Here again we have the growth when the length was about 12 centimeters. Stimulatory effect. Next stages was, what about if isolate the explants just directly from tulip bulbs without planting? So we have tulip bulbs, inside we have flower buds. What about if we isolate the explants? So this experiment was made October 27. If applied auxin or this kind of explant in level, we observed almost no reaction, no growth. So we applied to very early, after flower bud formation, no reaction on auxin. Next stage, we use partially cooled tulip bulbs and uncooled. In that case, it was the experiment made on November 23. We have control initial stage. We have stimulator effect, both in uncooled tulip bulbs and cooled tulip bulbs. Next stage, we use this just diagram of the same data presented, maybe next one. In that case, we applied explant isolated for full cooled tulip bulbs and uncooled tulip bulbs. Initial stage, in control, we have observed some growth, not so much, but with II treatment, we have the similar growth in uncooled and cooled tulip bulbs. So it is question why the reaction on IAA, both explant isolated from uncooled and cooled tulip bulb is similar. So I was thinking that in this case, it is not problem only of temperature, independent, it is 17 on 5 degree, but also the age of maturation, it is a very important factor. In both cases, it was after flower formation, it was about 4 months. But this phenomena needs fire study and to know the mechanism. Yes, here, if you keep also independent on uncooled tulip bulbs or cooled explants in light, on the darkness, results were similar. So this comparison of the treatment, uncooled in darkness, cooled in darkness, the growth is similar. After a treatment, dark one, similar effect. In the light, also similar effect. So our experiment, we use also another system, segments isolated from the bulbs. We use the leaf, stems, and the pistils. From cooled and uncooled tulip bulbs, we have here isolated the stems, pistils, and the leaves. And we use in our experiment, what about our... Yes, we found here you applied pistils from an isolated uncooled tulip bulbs to the medium containing gibberellin. We have observed strong stimulator effect, even September 23 of October 21. So it's mean the gibberellin have important role of the growth of the pistils, which is later source of the auxin. Here this pistil from cooled tulip bulbs. In this case, the gibberellin is much smaller degree stimulated the growth of the pistils, because during the cooling, naturally, gibberellin is increased in the pistils.